Hello students. Today we are going to discuss about transportation, biology, third lesson, 10th class. So what is meaning of transportation, right? So meaning of transportation is sending materials means like nutrients, oxygen. These are the necessary things required for any living organism, right? So these materials should reach each and every cell of the organism. Okay, so some in developed organism we have some particular organs which do this transportation work. If you take lower organisms like amoeba, what it will do? Amoeba have see here. Suppose amoeba is a single cellular organism, right? So we know it is a unicellular organism, right? So even though it is unicellular organism, it perform all the life processes. What are the life processes again? uh, Respiration, digestion, excretion, reproduction, transportation of materials, all this comes under coordination, right? All this comes under life processes. Here also, even though it is a single celled organism, this is nucleus, right? We know this one is nucleus, right? So, and cytoplasm present between the nucleus and cell membrane, this is your cell membrane. Okay, that is your cell membrane. Cell membrane. Okay, I'm not writing completely. Okay, what will happen if even though it is a single cellular organism, what it will do, it need oxygen, right? It need oxygen, and it need nutrients, right? So these should be taken by the membrane, and send it should reach. Suppose if it is taken from this part, so it should reach all the parts of the uh, this cell, right? So how they are moving? By the process of diffusion only. High concentration. What is diffusion? Particles moving from high concentration to low concentration, right? So see here, so even though it is single cellular organism, it is not having a particular organ to transport all these materials. Some current wo- cytoplasm will have some movements in it, Brownian movements, right? So those will transport the materials throughout the cell body. Okay, right. In developed organisms like humans and animals, they will have some special structures. Like in human beings, heart will send all the materials to the throughout the body. So these nutrients circulates throughout the body. They mix with the blood and they circulate with the body within the body. Here we have first topic is uh, observing the internal structure of mammalian heart. So lab activity, observing the internal structure of the mammalian heart. So what materials required here? We have to get a goat or a sheep heart. Okay, so remaining we need a sharp blade, right? Soda straws, or pen refills. We can use pen refills also, and a scalpel, tray, jug of water, decision, scissors, and forceps. So what we have to do before coming to the lab? uh, Wash your heart, the goat heart, thoroughly, so to drain the blood from the heart. So to make it clean, to remove blood from the heart, so wash it thoroughly. So if if you observe heart structure, if you observe heart structure, so what you will see generally, anterior portion is, right, anterior portion, lower part is called anterior portion, right, and upper portion is called posterior posterior portion. So what we have to do, stand it, make it to stand and cut it in longitudinal section. So vertical section, longitudinal means. So and observe the parts of the heart. So it is in pear shape, right? It is covered with the two membranes, and it is connected with some blood vessels. Large blood vessels are there. We see large blood vessels like aorta, pulmonary artery, we see that four blood vessels will be connected, and broader portion, upper portion is broader portion, and lower portion, anterior portion is narrower, right? It is in the shape of pear fruit. So this is our observation from this lab activity. Next topic is internal structure of the heart. Internal structure of the heart, right? So see here, first see the parts of the heart. Okay, first one. So if you observe internal structure, heart is having four chambers, right? It is having four chambers, and already we discussed upper portion is somewhat broader when compared with the lower portion, anterior portion and posterior portion, anterior portion, posterior portion. Sorry, what we discussed here. So I'm erasing all this. So this is your anterior portion. Anterior portion, this lower part is posterior portion. I'm writing half only, posterior portion, right? So it is broader at anterior portion and narrow at posterior portion, right? So it is in the shape of pear fruit. Every organ in human body is covered with two layers. If you take brain, heart, right? So these are covered with two membranes. These two membranes are filled with some fluids like structure, right? That it is called pericardial fluid. This membrane is called pericardium. The name of the membrane is pericardium. What is the name? Pericardium. And fluid is pericardial fluid. What is the use of this fluid? So suppose it protects your heart from shocks and jerks. Suddenly someone is hitting a person, right? So it protects. Rib cage is protecting your heart. If still uh, this membrane and the fluid acts as a cushion for the heart, right? Next, heart is divided into four chambers, right? So we call these two combinedly as atria. These two are commonly called as atria, and these two ventricles. So here you have to consider it as so this side is left side, here this side is right side, left side, right side, right? So from this we are getting oxygenated blood is going to lungs. From this blood is going to lungs, right? See here, pulmonary vein, we see parts of the heart. So this is, here we have four chambers: left atrium, left ventricle, right ventricle, two auricles, two ventricles, right? Two auricles, two ventricles. So this you have to write it as left auricle. Okay, left ventricle, left side. Right side, left auricle, right ventricle. Okay, from the, right, from the lungs, pulmonary vein. Before going to this topic, we should know types of blood vessels. In our body, blood flows in a closed circulatory system. It's not open circulatory system. Blood flow in the tube-like structures, right? Those are called blood vessels. Okay. Here in our human being, we have three types of blood vessels. Those are arteries, veins, and blood capillaries. What? Arteries, veins, and blood capillaries. Right. So arteries generally they carry oxygenated blood, oxygenated blood, right? So if you observe, walls of the arteries are thick when you compare with the veins, where they have very thin walls and they have thick walls. Why they have thick walls? Because arteries are originated from the heart. So heart is having some pressure, blood pressure. So whenever it is contracting and relaxing, so heart contracts and relaxes, right? At that time it will have some pressure on the blood vessels to withstand that that force. So they are having more thickness. When veins will not have that much of pressure from the heart, right? And they veins carry deoxygenated blood. Okay, veins carry deoxygenated blood, arteries carry oxygenated blood, and walls are thick to withstand the pressure from the heart. So here thin walls are thin, right? So in this veins you will find blood walls to stop the reverse flow of blood. We see valves in the valves, walls in the veins. Okay, right. Whenever human, right, so they are arteries originate from the heart, and they divide and convert into blood capillaries, right? So this turns into small tubes. So those are called blood capillaries. So if you observe, thickness of this blood capillaries is single cell thickness. So you'll see single cell thickness, right? Where you will see single cell thickness is whenever diffusion occurs, there we find this type of blood vessels, right? Suppose any tissue is present here, right? It has to send oxygen and it should send nutrients into the tissues, right? Or cells, and cells will take the materials and send out CO2 to the blood capillaries, and they send waste to the blood, right? So wherever diffusion occurs, there we find the blood capillaries. Each and every cell will get the nutrients from from these blood capillaries only. From how they are forming, they are coming from the splitting of or arteries. Whenever arteries are splitting, and whenever they are forming single cell thickness tubes, those are called blood capillaries. These blood capillaries again they rejoin, right? And they form veins, and they bring deoxygenated blood to the heart, right? So why we are discussing this is, see here, right? So already we discussed four chambers are there. Left atrium get the oxygenated blood from the heart, lungs, right? It is getting oxygenated blood from the lungs. Okay, next, then it is sending to the left ventricle. Then where it is going? It is going to the aorta, right? It is going to the aorta, and aorta is sending to all parts of the body, right? So aorta is a artery, right? So that is what it is indicated with red color. Blue color one is not this one, this part, only these two blood vessels and this one, right? From this superior vena cava and inferior vena cava, superior vena cava, inferior vena cava, they carry deoxygenated blood from all parts of the body. Superior vena cava brings deoxygenated blood from the neck and brain, head, right? From these parts it collect deoxygenated blood, and inferior vena cava brings, they collect anterior part, means legs, hands, our body. From that place it collects deoxygenated blood and send it to right atrium. Right atrium have two entries. One is from superior vena cava, second is from inferior vena cava. Then blood, deoxygenated blood reaches right atrium, then goes to the right ventricle. From this right ventricle it travels through the pulmonary artery, right? Here we have one question is that, now only we discussed arteries carry oxygenated blood, but this is different. It is named differently. Why? Because so it carries CO2 content blood, right? It carries CO2 content blood means CO2 carry, who carries CO2? Blood? Veins. But name is pulmonary artery. But when we are calling them as arteries, when they are originating from the heart, we are calling from they as arteries, even though it is carrying deoxygenated blood. So what is it is having? Structure of arteries, means if you observe thickness of this blood vessel is more as arteries. So that's what they named it as. Pulmonary artery is a special blood vessel which carry deoxygenated blood to the lungs. Okay, here. If you observe here, pulmonary vein, which is connected to lungs, right? So from the lungs, it is bringing oxygenated blood. It is bringing oxygenated blood, right? So now pulmonary vein is bringing oxygenated blood to the left atrium and it is named it as vein. Why? Because so whatever force coming from the heart will not come on will not work on this vein. And if you observe structure of this blood vessel is same as the vein. That's what this is a special blood vessel which carry oxygenated blood. So even though it is named as pulmonary vein, and the pulmonary artery carrying deoxygenated blood and the pulmonary vein is carrying oxygenated blood. So deoxygenated blood here, oxygenated blood. So it is going to lungs. From this it is going to lungs. From the lungs it is bringing to the heart. Okay, I think you understand the names, right? Next, we have to discuss about walls. What are the walls present, right? So see here. So between two chambers we have blood vessels, right? So walls. What are walls? Walls are the special structures which regulate the flow of blood. They are the special structures which regulate the flow of blood. Here we have from right atrium, right ventricle, tricuspid wall. Okay, and between left atrium, left ventricle we have bicuspid wall. This one, this portion exactly. Mitral valve, it is named as mitral valve, right? Not this portion is valve, right? So these are the valves we see here, right? This is bicuspid valve, it is tricuspid valve. This one, tricuspid valve, right? So if you observe tricuspid, bicuspid, uh, whenever you observe between these two, right atrium and right ventricle, it is named as tricuspid valve because three flap-like structures. Suppose here you will find three flaps, means generally it is having two flaps only, muzzle like structures only, which open like this and close like this. Open, open, close, open, close, like this. But here we find one more is there. Three layers will be there. Three flap like structures will be there. That's what it is named as tricuspid valve, right? Here only two flaps, only two will work like this. They open. Open means they open like this, and whenever they are closing they come like this. They stop the reverse flow of the blood. Okay, right. So that is, these are the two walls present between right atrium and right right atrium, right ventricle, and left atrium, left ventricle. Next, see here one more wall present between left ventricle and aorta, right? Whenever heart contrast, from the left ventricle blood enters into the aorta, right? So to stop the reverse flow of blood, so here also we have aortic wall, right, or systemic wall here between right ventricle and pulmonary artery. Between these two we have one pulmonary valve is present. Okay, pulmonary valve is present between these two, right ventricle and a pulmonary artery, that is pulmonary valve aortic valve or systemic valve. This is named as systemic valve also. In our book, it is as systemic valve. Right. Next, what aorta? Aorta is the largest blood vessel which starts from the heart. Right. It is turned into left side. Right. It turned to left side. Okay. Right. Whenever doctors take blood pressure, they check on the left hand, right? So most of the pressure comes to the left hand because of it is turned to left, right? So that is the reason. Next, what we missed? Right, aorta starts from the arise from the heart, and it is the largest blood vessel. It supplies oxygenated blood to all parts of the body. Upper part of the right ventricle pulmonary artery arises and supplies deoxygenated blood to lungs. From here it goes to lungs. So it is bringing blood from lungs to the heart. From here it is deoxygenated blood going to the heart. Right. Next, artery structure of heart is finished. Right. Next we have one topic is that. So next our topic is cardiac cycle, right? So suppose if the human heart starts beating around 21 days during the embryonic development. So 21st day, whenever embryonic zygote is forms thereafter, so it start beating, right? So if it is stopping means it result in the death of the person. So how heart functions? We'll see here. Suppose if you think no blood is first you I imagine that nothing is there in the heart. Everything is empty now. You think this left atrium, right ventricle, left ventricle, all are in empty stage. Right? Now pulmonary vein carries oxygenated blood from the lungs. Right? You have to imagine that left atrium, right atrium are receiving blood at a time. Means it is getting deoxygenated blood from superior vena cava, inferior vena cava, and it is getting from oxygenated blood from the lungs to the left atrium at a time. Right? Now these two are filling. Right? Now, they whenever blood coming from the pulmonary vein and superior vena cava, inferior vena cava, these auricles uh, start taking the blood, right? That position is called resting. Right? Whenever it is chamber is filling, that position is called resting. Right? Next, this stage is called relaxed stage also. Relaxed stage means diastole. After receiving blood, so it should send blood to the right ventricle and left ventricle, left atrium to left ventricle, right atrium to right ventricle. What? So when it starts contrasting, so whenever it starts contrasting, blood from this auricle squeezes into the ventricles. Okay. So right now these two are contrasting, ventricles are relaxing. Auricles are contrasting, ventricles are relaxing. So here these two are in systole phase, these two are in diastole phase. Relaxed stage now, these two are getting blood from the arteries, sorry, auricles. So these two are in now relaxed stage, these two are in contrasting stage. Right? Now blood is coming from auricles to ventricles. Right? In the next stage, after filling the blood in the ventricles, these ventricles starts contrasting. Okay. So whenever these two start contrasting, blood enters into the from left ventricle to the aorta, like this it will go from the left ventricle, then it goes to the aorta, oxygenated blood. From the right atrium, right ventricle is receiving deoxygenated blood, then it is going to the pulmonary artery and to the heart. Right? Next, so from here we have a chance here. If you imagine that there are no walls here present here, blood may go reverse also. To stop the reverse flow of blood, these two walls, what was tricuspid wall, bicuspid walls, they closes very sharply with some sound, high sound. Right? They whenever ventricles start contrasting, these two walls closes very sharply. That time we hear one sound, that is lub. we have lub dub sound, right? One is high, little, second one is somewhat low, right? So that sharp sound comes under lub sound, right? Next, these two walls, from the walls, blood enters into the aorta, and from the right ventricle to the pulmonary artery, right? Next stage, after emptying these two ventricles, now whenever these are contrasting, these two auricles will get the blood from the lungs and the superior vena cava, inferior vena cava, right? So whenever these two are contrasted, these two are in relaxed stage. Okay. Next, after contrasting, again these ventricles starts relaxing and getting blood from the left atrium and left ventricle. Right. Here, if you observe, there may be a chance of reverse flow of blood from the pulmonary artery and the systemic wall. Right. From these two blood vessels, blood may take reverse flow. To stop the reverse flow, these two walls closes at a time. That time we hear the second sound, that is dub. So first sound is from the tricuspid wall, bicuspid wall. Second sound is from these two walls, right? So in this way we get, so these two auricles resting means they get the blood from the pulmonary vein and superior vena cava, inferior vena cava, right? Whenever these are resting, these ventricles will be in contrasting position, right? So whenever these two are contrasting, blood may to stop the reverse flow of blood, tricuspid wall, bicuspid wall closes very sharply with some sound that is called lub. Whenever this closing, that time we get dub sound. I think you are understanding this cardiac cycle. Next topic we have is single circulation, double circulation. Single circulation, double circulation. Right. See here, if you observe blood circulation in a fish, so how fish have gills. From the gills, it takes dissolved oxygen from the water into the gills, right? So from the gills, uh, by the process of diffusion only, blood oxygen enters into the gill capillaries, right? So capillaries means they help in the diffusion process, and we know the structure of capillaries. They are having single cell thickness. They are having single cell thickness. Next, so from the gills, so it will not send blood to the heart directly. It will send oxygenated blood to all parts of the body, right? So here capillaries are splitting, right? It is sending blood to the all parts of the body. From the body parts, right? From the body parts, it is collecting, veins are collecting deoxygenated blood. It is collecting deoxygenated blood, right? It is sending waste to the waste deoxygenated blood to the heart. Then this heart is pumping that blood to the gills again. So what in our heart, what will happen? See, we will see, right? First, whenever we are getting oxygenated blood, so here lungs capillaries. So we know alveoli, from the blood capillaries, there is a continuous exchange of oxygen and carbon dioxide, right? From the lungs, blood capillaries takes oxygen, right? That oxygen mixes with the hemoglobin. There it forms oxyhemoglobin, right? That blood comes to the heart. This heart pumps the blood to, from, right, where it is reaching left auricle, then going to left ventricle, then going to aorta. Aorta is sending all parts of the body, right? Okay. Next, what is happening in our body? So if you suppose think it as this is your body. From, so blood capillary, aorta is splitting, and it is forming blood capillaries, right? Where you find blood capillaries? Everywhere in our body, wherever cell present, there we find the blood capillaries to exchange the nutrients and oxygen, carbon dioxide, waste, everything, right? Cells take oxygenated blood and nutrients from the blood, oxygen and nutrients from the blood, right? And they send out waste and CO2 into the blood capillaries, right? These blood capillaries slowly forming veins, right? These veins, inferior vena cava, posterior vena cava, this comes into the right auricle, then blood, deoxygenated blood going to right ventricle, then going to pulmonary artery, then reaching to the lungs, then it is reaching to the right right auricle, right ventricle, pulmonary artery, then going to the lungs, right? So if you observe here, blood is circulating through the heart two times. It is traveling through the blood, through our heart two times. If you observe this picture, so it is only circulating one time through. It is crossing the heart one time only. In our human beings, it is crossing two times. Oxygenated blood coming to the heart, then going to all parts of the body. Deoxygenated blood collected and sent it to the heart. Then again it is pumping blood to the lungs. Right? So it is crossing two times. Here it is crossing one time. So it is called single circulation. Here we call it as double circulation. So blood flows through the heart only once to complete one circulation. This is single circulation. Here, blood flows through the heart twice for completing one circulation. So it is called double circulation. Here it is double circulation. Next, lymphatic system. Lymphatic system. Right. Whenever you are traveling in the bus, so what will happen? So if continuously you are traveling in a bus, for throughout the night, morning by morning we are reaching our destination. Whenever you observe at that time, so you find that your legs are swelled, right? So why they are swelling? Okay, because some blood is or something is there in your legs, right? So after some time, whenever you after walking some time, they come to normal position. Why, we will discuss here, right? So blood circulates in the blood vessels, right? Pushed by the heart. From the heart it flows into the arteries. Arteries are splitting and converting into blood capillaries, which helps for the diffusion, right? So after diffusion, right, next it takes oxygen, nutrients, then it send out CO2 and waste into the again those blood capillaries, right? Next, to supply nutrients to the cells, thus liquid portion, see here, the liquid portion of the blood with the nutrients flow out of the capillaries. This is called tissue fluid. Right, I'll make you to understand that. Suppose if you think that blood capillaries are going like this. Suppose you have numerous cells which are taking oxygen and nutrients. They are taking nutrients also, right? What they were giving back, CO2 and waste also sending, right? So whenever these materials, CO2 is a and dissolved carbon dioxide, right? And waste, whenever these are passing into the blood capillaries, some or this fluid is called tissue fluid. Okay, right. Whenever they are crossing, this fluid flowing into the blood capillaries, fluid may leaked into the spaces between the cells. Okay, this fluid flows through the system called lymphatics. Whenever these two, waste and carbon dioxide, flowing in the form of liquid only, so whenever they are entering into the blood capillaries, some portion may leaks from the cells, right? Again, one more system is there which will send these materials into the blood capillaries. That system is lymphatic system. Lymphatic system, it is a system which is parallel to the veins. Wherever veins are there, there you will find lymphatic system, right? Lymphatic system is parallel to the veins. Okay, lymph is a vital link between blood and tissues, which is essential substances pass from blood to cells and excretory products from cells to blood. Suppose whenever nutrients and oxygen are exchanging, something may leaks, right? So this is also sending back to the blood capillaries through the lymphatic lymphatic system. Lymphatic lymphatic system, right? These are the parallel to veins, right? So the muscles which are attached to the skulls, okay, how it is, you are telling, again they are sending material to the veins. How it is possible? Whenever you are moving and whenever you are walking here and there, from the bones and muscles you will get some pressure, right? With that pressure, these lymphatic vessels send the waste materials into the blood capillaries. Waste or useful, everything they send into the blood capillaries, means leakage fluid is sending back to the veins, right? Next, blood is a substance which contains solid liquid particles. Lymph is a substance which contains blood without solid particles. So it is like only just blood, only plasma will be there. So nothing solid particles will be there. Tissue fluid is a lymph present between tissues. Right? This liquid portion after formation of blood clot is called serum. Muscles which are attached to the skeleton acts as pumps when they contract and helps in pushing lymph flowing into the lymphatic vessels and blood flowing in the veins towards the heart. Next, evolution. Evolution, right? So evolution of transportation system. Right. We know every organism came from single cellular organism, right? So amoeba, how it is sending. So we already discussed it, it is in the process of diffusion, right? So in the cytoplasm we have some movements, right? So it is having some natural currents in its body. So because of that, it is this cytoplasm will have some movements. Those movements are called Brownian movements. Brownian movements. Okay. After that, if you take sponges, suppose if you observe sponges, suppose one rock is there, sponges are attached to the rocks, they will not move, right? They and if you observe the body of the sponge, so it is having so many holes through the body, right? How it is helping? Whenever seawater comes and leaves the body, right? So they take nutrients from the water and oxygen from the water, they send, water flows through this pores, right? And whenever this water is passing through the pores, they take nutrients and oxygen, right? Oxygen and nutrients into the body and send CO2 and waste into the water. They are sending waste, right? So it is having one tail-like structure, flagella. We call it as flagella. What it will do, particular time after 1 second, 2 seconds, it will continuously moves its flagella, means it's a trail-like structure. So it will move this flagella and then it will get some movements in its body. With that movements, it is taking nutrients and oxygen, sending waste and CO2 into the water, right? Next, we have hydra and jellyfish, they comes under cnidarians, right? They are having gastrovascular, hydra and a jellyfish, their body having gastrovascular cavity, so which will have taken up the function of digestion and transportation nutrients to each and every cell of the body. If you take your jellyfish structure, so it will have like this. So it is having gastrovascular cavity throughout the body. It is sending directly to the cells. It is sending nutrients and oxygen to each and every cell directly, right? Next we have platyhelminthes, right? So they have highly branched digestive system. Each and every system will get the, suppose any organism is there, it is having digestive system spread throughout the body. So it is not located in one particular place. So it is branched throughout the body. Each and every cell will get directly food from the digestive part. Okay, next we have roundworms. So pseudocilium has taken up the function. In roundworms, we see the pseudocilium has taken up the function of collection and distribution of the material. So what is uh, transportation system you find in roundworms is pseudocilium. Annelids, one pulsatile vessel will be there which transport the blood to all parts of the body. Right, here onwards, pulsatile organs are started where annelids. Right, next if you see arthropods, heart. So you find here heart in these animals, they pump the blood to all parts of the body. Right, next we have blood pressure. Blood pressure. We know that diastole means relaxing stage of heart rate, relaxing. Systole, contrasting. Right, right. Relaxing means they are getting the blood from the pulmonary vein and pulmonary artery, oxygenated blood and deoxygenated blood into the arteries, auricles. Then blood is going to the ventricles, right? So whenever they are sending blood to ventricles, that stage is called contrasting, right? Whenever their ventricles receiving blood, that time stage is called relaxing. Whenever ventricles are contrasting, that is called systole, diastole, right? So here how they measure the blood pressure. See here, this instrument is called sphygmomanometer, right? Sphygmomanometer. So generally they take readings on the left hand. We know because of aorta starts turns because of aorta only it is taken from the left hand, right? So what are the measurements of your systole and diastole? Right, whenever ventricle starts contrasting, right, we see only, suppose if you see this is as your heart, we are having left auricle, left ventricle, left left ventricle, sorry, this is right auricle, right ventricle, right auricle. Okay, right, left auricle gets oxygenated blood from the lungs, right? Next from the body parts it is getting deoxygenated blood, right? Next whenever these auricles are getting blood, this position is called relaxing, right? Next whenever these two are relaxing, these will contrast, right? Right, next whenever after relaxing stage, from the auricles blood comes into the ventricles, right? So they squeeze the blood into the ventricles, then ventricles receive the blood, then ventricle starts contrasting, they send blood to the aorta, from here to here aorta it is sending to the pulmonary vein, right? So you see high pressure, one generally we get this from this ventricles contraction and relaxation only. Whenever these ventricles are contrasting, that time we get 120 mg readings in sphygmomanometer. Okay, right. Whenever after pumping blood to the aorta, these two ventricles are emptied and they start relaxing. So that time one measurements of pressure during the time blood is forced out of the ventricles, this is called systolic pressure. So whenever these are contrasting, that is called systolic pressure, right? And a second reading is taken during the resting of the ventricles. Resting means they are getting the blood from the auricles, that time means at the particular stage we get high reading, that is 120 mg one time, we'll get very low reading 80 mg, thereafter again we get high sound in the sphygmomanometer. So these two readings are normal person BP, blood pressure. Okay, next we have coagulation of blood. coagulation of blood. So what is happening? So when you get an injury, blood flow out of the wound for only a short time, right? It is not going. Suppose any scar is cut is happened on your hand, what will happen? So within 1 2 minutes, it stops bleeding, right? Then after some time, this wound also somewhat closed, right? So it is not going continuously outside. This cut is filled with reddish solid material. This solid is called blood clot. Okay, we know this is blood clotting. If blood is did not clot, what will happen? Blood flow out of the body continuously. Even anyone with even a slight wound bleeds profusely, right? So because of coagulation blood only, coagulation of blood. So this is happening in our body, right? How it works, we see here. So I'll write one thing. Thrombin, fibrinogen, fibrin, activated by thrombokinase, thrombin. Just we'll discuss this only, right? When blood flows out, what will happen? Whenever any cut is happened on your hand, something is there happened to your hand, so blood is coming out of your hand. So what will happen at that time? So we know platelets are one of the reason for blood clot, right? So platelets only acting here. Platelets release an enzyme, right? It is release an enzyme called thrombokinase. First material. So first platelets acts on platelets acts on thrombokinase. Where you find this? So it is dissolved in the blood. All these materials are dissolved in the dissolved stage. Normally it is there in dissolved stage or inactive stage, um, right? So which platelets release an enzyme to, called thrombokinase. It is releasing one enzyme is called thrombokinase. So it is working on. So thrombokinase acts on another substance present in the blood called prothrombin. So now it is acting on thrombin, right? Converting into thrombin. So prothrombin, where it is present dissolved stage, again it is present in the blood in the dissolved stage, right? Inactive stage. So it is converting into thrombin. Thrombin acts on one more substance that is called fibrinogen, present in the dissolved stage, again converting into fibrin, right? Blood cells entangle in the fibrin fibers forming clot. Whenever suppose any cut is opened here, any one cut is there on your hand like this, so they entangle it this place. They form uh, thread like structures. They pull the both the sides and they make one close thing, clotting. So it's form clotting, right? The blood cells entangle in the fibrin fibers forming the, so it makes it close and they make clot. The fibrin fibers attach to the edge of the wound and pull them together. The yellowish straw colored fluid portion after formation of the clot is called serum. Okay, right. So how to remember this one? If you go in a reverse way, it is easy to remember. Fibrin. So we find after cutting or anything happened to your hand, near the cut we find this thread like structures, right? They are pulling. So if you take it as fibrin, right? So fibrin comes from the fibrinogen, which is activated by thrombin. Thrombin is one more substance which is coming from the prothrombin. So this is activated by thrombokinase. Thrombokinase is an enzyme that is coming from the platelets. Writing is different. Remembering purpose only I am telling this one. So everything we cannot remember. No, everything goes with a logic. So this is the logic to remember. So just to remember this word, fibrin is coming from fibrinogen, which is activated by thrombin. Thrombin is one more substance coming from prothrombin, which is activated by thrombokinase. Thrombokinase is an enzyme released by platelets. Okay, right. Next we have root pressure. Right. Here we will see root pressure. What is happening root pressure? Already we know that we have two new words, osmosis. Before going to this, we'll discuss one topic, diffusion and osmosis. These two works in opposite direction. Suppose diffusion means particles moving from high concentration to low concentration, right? We know this one. Particles are moving from high concentration to low concentration is called diffusion. Here in osmosis, particles move from low concentration to high concentration, reverse of your diffusion. Suppose here materials are very low in what soil, materials are very less, but in plants maybe content is high concentrated. So here actually in the process of diffusion they should go into the soil, but from the soil minerals are taken by the plant, right? So that is because of osmosis process. We'll see this one. Right. What is happening? So osmosis plays an important role in movement of water from root hairs to the xylem. What is root hair? So if you see sprouts or a small baby plant, there you will find some root-like structures, right? If you see here any plant, here you will find root-like structures below the soil, and on these roots you will find fine threads-like structure, right? Those are called very thin structures. Those are called root hair. So whenever you observe under the microscope, you will see like this. So only this part is your root hair, remaining all is soil part. This all comes under soil particles only. Only this part is your root hair. Okay, right. How it is working, we will see. Right. Suppose if you observe this vacuole of the root hair or cell sap, after vacuole we have one more is cell sap. After that, so outer cells are present, then soil is present outside the cell sap and vacuole, root hair. Okay, this everything is root soil, and it is having some moisture, material, minerals, right? Everything is dissolved in the water. If whenever water is entering into the root hair, minerals also travels into the root hair, right? So if you think that here concentration is low in the soil, what low? Minerals concentration. I am taking minerals concentration is very low now, right? So if you already these are the sprouts, I am talking about sprouts. So what materials will be present in the roots? Somewhat they are having high concentration. Here we have high concentration, right? What will happen? So I am taking water. I am talking about the water present the root hair. So these water near the root hair are very diluted, means less concentrated. Here we have more high concentration, right? Now because of that, water will pass into the vacuole by the process of osmosis. Into the vacuole, water is entering from the soil. From the soil, water is going into the cell sap, then entering into the vacuole. Then slowly, not only we'll have only one hair, so many numerous root hairs will be seen on the uh, roots, right? So all these work at a time, right? So when all these are taking water from the soil, means because of osmosis water is entering into here, right? Now water is entered here. If you take one more cell here, suppose if you observe one more cell is present, two cells are present, like this we'll see. Now water is entered into the first cell. So from here less concentrated to high concentration, whenever this cell is getting materials, it water also coming into the cell. This cell also more diluted. Now it is more diluted than the neighboring cell. Now if you take these two cells only, it is high concentrated, low concentrated. Then watering entering with the neighbor cell. Then if you compare these two, high concentration, low concentration. Again particles are water is moving to the nearby cell, then going like this, and they reach the vacuole and enter into the xylem. Okay, right. So water passes into the neighboring cell, which is in turn becomes diluted. Finally, water enters the xylem vessel. xylem vessels. As there are vast number of root hairs and root cells involved, pressure in the xylem vessels develops. So as water enters into the next, these are entering, they check if osmosis going on continuously, they check each and every cell concentration and water enters into the next cell, right? So by this way water comes into the root hair. So with this process, pressure created in the xylem, so it is a continuous process, water is coming from the cells to the xylem. Okay, so this pressure is called root pressure, right? So when a pressure in the xylem vessels develops which forces the water upwards, this total pressure is known as root pressure. Whenever they are asking about how explain about root pressure, means you have to write this activity, not activity, about this topic. And we have one simple lab activity about root pressure, that is, take a potted plant, right? So already watered this plant, right? Next time what I am doing, I am cutting the stem of the plant, inserting the glass tube. Before that, glass tube width should be equal to your stem width, then only we can fix them easily using rubber cork. Okay, rubber tubing to stop the reverse flow of water. So what I am taking first, what I did now, I cut the stem, I inserted the this rubber tubing. So here nothing will flow into the pot. So I am pouring some water here. So now up to here only I poured my water. So I named it as M1. I marked it as M1 and I kept this arrangement aside for 2 to 3 hours. After some time you observe that water levels are increased. Water levels are increased to M2, right? So even though there is no plant, water level increases in the tube. So measure these measurements. So first level, we are before that only we are pouring some water, right? So second time water level increases to M2 because of how it is, from where this water comes from the roots, water is entering into the stem then entering in the test tube. Water levels are increased because of root pressure. This is your activity, lab activity, simple lab activity. With this your topic complete, Asha.